Where is it? Here it is. Okay. All right, there you go.
Thanks so much. Okay, I'm a little late presenting, but I was sick last Monday, and that's kind of whenever Dr. Yost like gave us all of our possible like pathogens too. And so I like went into the doctor and was like, I might be dying. I don't know. I need you guys to tell me I'm fine, which I was fine, but so I picked necrotizing fasciitis. I actually am super excited about this. And yeah, so this is going to be a rare, life-threatening, rapidly spreading bacteria. It infects your soft tissue, which is below your like skin, but above like your muscle. It's in that like connective tissue or your collagen and all of that is. There's 4 different kinds that are gonna be associated with it, and all that means that is you have 4 different classes. The mortality rate is anywhere from 20 to 80%, and all that does is just depends on your health beforehand. It could be not a big deal if you're somebody, 20 years old, perfectly healthy, no issues. But, if you're 85 years old and you have a lot of pre-existing conditions, a lot of immune suppressant issues, you are going to be more towards that 80% side of it. And then the key early diagnosis is the key to preserving tissues and basically your life. This is illness that spreads pretty fast. It's gonna break down your tissue really quickly. So, you want to try and get in and get treated as fast as possible. Back to anatomy. Here's a little like diagram of where we're at with the skin and the different layers. So like with here, you just have, like I said, your muscles here, and then your subcutaneous fat, your dermis, and your epidermis. Generally speaking, like I've done hair for 10 years. So like for me, I'm only allowed to treat like the epidermis, your top layer. If you want to be like a nurse injector, you have to get your RN license and you're allowed to inject into this dermis layer. That's important because in a clinical setting, say you do get a deep cut or wound and it gets to this dermis layer, or even close to that epidermis where you have your capillaries and nerves, you can get infection in there really quick and really fast. And this is all within like a 24 to 48 hour timeline here. So the infection comes in and then it spreads really rapidly and shuts down all of the capillaries, the nerve tissue, and that's not good because that is how you get, blood flow and like your immune response to these issues. So this is just a really quick breakdown of all the different classes. Type 1 is your polymicrobial. All that means is when you take a swab of somebody, they're going to present with multiple different types of microbes, usually about 4. The type 2, this one's going to be your most common, and it's going to test positive for your group A, Streptococcus aureus, and also presents with MRSA as well. Type 3 and type 4 are pretty rare. They don't happen often, especially type 4. You have to have like have all the bad things happening. Like you have to have a pretty good compromised immune system, like and just have like a huge surgery or huge like wound done, have a lot of that like underlying tissue exposed for more of that fungal infection to present. So your virulence factors, there's a ton of them, and I just did the 2 for the most common bacteria that you're going to find. Generally speaking, both of these have proteins, toxins, and enzymes that are going to inhibit like phagocytosis. They're going to present toxins that break down the red and white blood cells and overstimulate the immune response, which is from the LPS, the lipopolysaccharide. Last word. The LPS from the blood cell. You're also going to have enzymes that have like tissue damage that break down blood clots. And all the things. And the other thing that's really big about this, especially MRSA, Staphylococcus is really good at learning how to become resistant to bacteria. So that's why MRSA is resistant to methylcycline, I believe. So this I have been so excited about. My husband, I've presented this to him like 800 times. He's over it by now. But so your risk factors, like I said before, I mean, the biggest risk factor is if you have an open wound and, that staphylococcus or streptococcus gets inside of it. Most healthy people, you're going to have both of those bacteria growing anyway, but like you have an overgrowth of them, and 9 times out of 10, it happens from a hospital setting. Explosion at subdermal tissue, diabetes is a big one too. So this patient presented, she is a 69-year-old female. She presented 6 months prior to the emergency room and was having a lot of like issues and, wounds that just wouldn't heal on the back of her neck. And she goes into the emergency room and her doctor is just like, yeah, I think it's just diabetes and tachylitis. Like, you have a history of diabetes, like, here's some antibiotics, follow up with us in 3 days and, we'll go from there. She takes the medication, nothing changes. And she's like, man, whatever, I'm not going back. And 6 months after, this is where we're at. Image A, she presents the emergency room and she has obviously this tissue that's just completely dead here. It has no circulation, it's red, arrhythmia, that tissue is just dead. This is a side CT of her. You can see all of this like inflamed tissue right here. That is essentially her tissue that's dead. These little black pockets are pockets of gas. And what blows my mind is like the fact that none of this infection got into her like CNS, and like that could have killed her so fast. So the only way to like effectively treat this is going to be debridement of the tissue. She actually did not know she had diabetes. So she came in, they noticed the sign. They immediately started her on a broad spectrum antibiotic. They started treatment for sepsis, blood and swab test for S. aureus and strep, came back positive. She was hospitalized for 2 weeks. And then after that 2 weeks, or throughout that 2 weeks, she had 4 debridement surgeries. A mode of transmission, IV drug use, insect bites, skin abrasions, and then abdominal and perineal surgery. This is a different patient. She had diabetes, did not receive the proper education for wound care for her feet, and presented to the emergency room. She had a 15-centimeter lesion gash on her foot, which I didn't know how big 15 centimeters was. I converted it. It's almost 6 inches. You can see that her tendons and bones are exposed there. They went through, did MRI, CTs, and on image C, that big round circle is going to be her tibia, and the smaller one is her fibia. Those little like black gas pockets are literally bacteria putting off gas, and it has nowhere to go, so it's just trapped in there. And then in image E, all of this like foul colored tissue here is literally supposed to be just normal white and red. All that's just bacteria. Afterwards, they took a histology slide of it, and that's what these are here. You can see all those like gas pockets. So you have your early diagnosis, you're just gonna have like red skin, it's almost gonna look like a sunburn. Midterm diagnosis and get treated. So this is probably a patient who was treated after, you know, within 24 hours of onset, maybe 36. And then this last patient here, they were diagnosed very late, obviously. I think that diabetes may have been at play here. Signs and symptoms, you know, I mean, it's kind of the same thing. You're going to have a lot of like redness. The biggest thing for this is symptoms is like you're going to see a patient present and they're going to have like a red lesion on the skin and it looks like a sunburn, but like the pain that they are going to have associated with this is going to be like unproportionable. You have your different diagnoses, which are going to be gangrene, cellulitis. Your diagnosis or diagnostics, you have your MRI scan like we saw earlier. You can look at your MRI and see the presence of fascia thickening and gas present. Your deep tissue biopsy, after you take some of that necrotic tissue out, they'll send it off to lab, they'll freeze it, and then look at it underneath the microscope and look for inflammation, you know, skin cells that are dead. You have your Lernick testing. This is just a scolding value based off of regular values. And if you have a score of 6 or higher, it is going to present that you probably do have a necrotizing infection of some sort. The way that you narrow it down is you are going to want to, after you have a sepsis treatment going on your patient, you're going to take this into the gram stain laboratory. And if it tests positive for group A strep, like, pretty good that like, yes, that is what we've got going on here. Clinical approach. So when necrotizing fasciitis is expected, like I said, or suspected, you're gonna throw a broad spectrum antibiotic at them, get them immediately in care, gram stain that bacteria. You're not going to want to waste time with this either. So like, you don't want to sit there and like wait for your testing and stuff to come back. You kind of want to get an early start on this, and debridement really is the only way, like removal of that tissue. And some patients have to have like surgery daily for weeks on end to remove that tissue, just depending on their mortality rate, essentially. Then after you have all of your surgery, you can come in and talk with a plastic surgeon and have all of this rebuilt. Prevention, I mean, pretty much washing your hands, making sure, that you're not going to allow any bacteria to overgrow. Whenever you have an open wound, this is why they tell you not to go, like swimming in a freaking lake or, hot tubbing with your besties, because like that bacteria can go into that wound. And then, within 24 hours, you have a big, huge, like tissue debridement. And there you go. So, I could ramble, however.
We appreciate that sort of like case study approach, including the pictures that are in there. Something I'm probably going to include more of in my future slides when I make edits, because the pre-made ones from McGraw-Hill just don't have enough of that. All right, so let's jump right into it for our cardiovascular system and lymphatic system diseases. First of all, most of us are aware of what the heart is and how it works and what it does for our body. So in our circulatory system, our heart is going to pump our blood. And remember, if you learn anything from anatomy and physiology, that with your arteries, that's just blood, essentially going away from the heart, or veins going back to the heart, essentially. They're carrying our oxygen and our carbon dioxide waste. So we also have the capillaries where we have like sort of the mixing of the two, where we're delivering oxygen to our tissues as well as getting back our carbon dioxide waste. The lymphatic system, we've learned about this already in our chapters 15 and 16. So these are lymphatic vessels, lymph nodes, our spleen, and our gulls, malts, and salts, and everything else are associated lymphoid tissues. All of that being tissue essentially where we have our white blood cells waiting to screen out any possible, bad guys or evidence of bad guys when that fluid is working its way back to the heart. So for our defenses for the cardiovascular system, it's obviously going to be housing a lot of your white blood cells. I mean, all of your white blood cells are going to be circulating through the lymph and the bloodstream. That's how they move around in the body. So that's where most of your defense is going to be. And it's keeping that area sterile. So it should be sterile, just like your CNS and your peripheral nervous system. This should be sterile. But here, instead of immune privilege, we have strong immune response. So all of your white blood cells and everything is there actually doing full on work of this, because we cannot have pathogens being disseminated to every single part of your body when you have an infection that becomes quite serious very quickly. So we have just a whole ton of those. So back to these terms, we've mentioned these previously, but just to summarize them again so that they're fresh in our minds, because almost all of this is going to relate back to that. Viremia is when you have viruses. Usually these are the ones that are going to be causing the meningitis, and that's why they're in the bloodstream, is because they're kind of transferring into your nervous system. So they can cause meningitis if they're found in the blood. If they're found in the blood, it's called viremia. Fungemia, fungi in the blood. Bacteremia, any presence of bacteria in the blood. And if those bacteria are flourishing and growing in the bloodstream, like an actual infection of the bloodstream, then that is septicemia, which can lead to shock and severely decreased blood pressure and eventually death, as well as disseminated inflammation. We consider this a closed system because the blood itself isn't leaving the area. Sometimes we have the fluid and some of the white blood cells will leave, but they will re-enter back into the circulation of everything. Our first example of cardiovascular and lymphatic disease will be COVID. Like we've said previously, everybody likes to think of COVID as being a respiratory disease because we think of it as being transmitted by the respiratory route, but it by far does not stop there. This virus can infect pretty much any tissue in your entire body and is disseminated by your circulatory system to do so. So it's able to recognize a ton of different, like it's a couple different receptors it can recognize, but those receptors are found all over all different tissues throughout the entire body. So we consider this a cardiovascular disease. It does present with some respiratory symptoms and can lead to secondary respiratory infections associated with it. This is a completely different virus than SARS. Okay, I had a student in one section who has picked COVID, and I have another student in another section who has picked SARS, and the poor student that picked the SARS virus does not understand that is a different virus. It is. COVID is caused by SARS-CoV-2. We're used to hearing about this, and SARS is caused by SARS-CoV. It was the first. It had outbreaks back in the early 2000s, and if you were alive for that and old enough to live through that, which I was, there was a severe fear that it was going to spread outside of China and be this deadly disease and all of that. But it went away. We had outbreaks of SARS through 2003, and then we never heard of it ever again. It disappeared. And then we got COVID, a completely different virus that was much worse. And so our fears were founded, but not for another 20 years later. So I just like to reiterate that because they are completely different. Are they related to one another? Absolutely. Absolutely they're related. And probably COVID came from genetic manipulation of SARS. If we're being realistic here, right? I don't want to get into conspiracy theories because that does remind me, I just want to be sure for those of you who do not keep up with the news, now is a time where you might want to, because I'm not talking about diseases. I'm just making a little stem of maybe keeping up with the news, because if you don't know what's going on with Ukraine and Russia right now, you should. And I'm going to be completely serious about this. We're probably going to war soon.
Us?
Yes, like I'm completely serious with Russia. Yes, and North Korea, to keep this clear. I'm not kidding. We just provided long distance missiles. We provided long distance missiles to Ukraine. I'm not saying that's a bad move. I'm just letting you know. Putin is not happy about this, and he has troops in the area where these long-range missiles were sent from North Korea. You're up to speed now. Just letting you know, if you're not scared about this, this is the time to not brush that under the rug. You should stay
We're
aware.
not talking nukes, right?
No, not nukes, long-range missiles.
Aren't
So-
there nukes involved? I don't think.
We aren't the only country involved in this, right? But Putin has made a message that he has very explicitly stated, NATO and the United States are in trouble. So,
Putin is stinky.
huh? Putin is stinky?
Yeah.
Putin got elected president. They had turned to democracy after they got rid of communism, right? They turned to democracy and they modeled their president voting system after ours. They have like a 4-year term, and then you can get elected for another term, and then whatever from there. Well, whenever, gosh, you guys, I'm a big fan of Putin as a person because he's ridiculous and he does have power for a reason, but I'm not a fan of like that side of it, like the political side, okay? But anyway, anyways, so when he took over from Boris Yeltsin and became president, he served his term. And then whenever he finished his term, his good old buddy, who was the prime minister, became the president for one 4-year term and changed the law so that presidents can serve for as long as they would like to, as long as they have support from the people. And then, so after Friend's little 4-year term was up as the president, and by the way, while this was happening, Putin was the prime minister. Putin was reelected as president, has been president ever since. This is since 2000. 24 years, basically, ruling Russia in a democracy.
Why
It's
do
a democracy, just to be clear. Does it look like a democracy to anybody in here? Does it look like a dictatorship? Because it should.
people still support him?
Do they have a choice? It's not like America, man. There's not real voting there.
Right.
It's really corrupt. Anyways, I only brought that up because of the conspiracy theories of SARS being manipulated by China as bioterrorism weapons, which they probably were. But anyways, that just reminded me of that. Stay in the news, my friends. Stay safe, stay aware. Okay, moving on to cheerful things. Let's talk about deadly diseases now. Yeah. Anyways, so again, like I said, this virus can infect all sorts of animals. It can infect all sorts of cells in those animals. And that's why we are considering it a cardiovascular disease. Other than that, I mean, it's pretty straightforward. We know COVID, we have had mutations since the original virus got out. We've had the alpha variant, which is more contagious than the original one. Delta, which is twice as transmissible, so meaning even more contagious than alpha, and it's more deadly. And this is the one we're most used to hearing about, COVID Delta, the Delta variant, and Omicron variant. Omicron has dominated North America pretty much ever since it made its appearance here in 2022. It's equally as bad as Delta. And we might sit here saying, then why aren't people dying as much? It's so much worse because of the vaccine. That's why. So we have that to thank for that. Of course, we have respiratory symptoms. Cytokine storm is also common in certain individuals, just depending on how your immune system reacts to this. You have an overreaction of those cytokines. So if you guys remember what cytokines are, they're just signaling molecules that help signal the immune response. And the immune response is what's going to form your inflammatory response. So that cytokine storm leads to hyperinflammation, and that's what's going to put you in the hospital. It's also neurotropic. As far as it can infect the nervous system and have nervous system effects like that loss of smell and headache and things like that, and can even lead to dementia and things. And up to a third of infections are asymptomatic. They say up to a third. This is the data that we have. This is how you find out having people come in and get tested regularly. But now that we don't have that, how many people are walking around carrying COVID that don't have symptoms probably increased since we had introduction of the vaccine. So most people don't have serious symptoms now after introducing vaccine. We have already learned about the vaccines that were introduced for this, so I'm not going to go into that again. But the best way to prevent spread of this was social distancing, masking, not going out unless you had to, washing your hands, and all of that sort of stuff. My husband and I had this discussion. But he was like, women don't wash their hands in the bathroom. And I said, no. That has not been my experience. I don't know about, okay, occasionally we see a woman, right? That might not. But he said, well, whenever we had COVID, Men started taking longer in the bathroom, but women didn't. They just weren't washing their hands. Like, men had increase in washing their hands and the women didn't. I said, no, sweetheart, that's because women already washed their hands. That's why. I mean, most of us know that most men, if they are just going into urinate, most of them probably at least weren't washing their hands. And we don't have to get into the descriptions and everything, but you're not going to fool me. You're not going to fool me with that. Okay. Endocarditis, this is when we have actual inflammation of the inner lining of the heart. So it's usually spread from some other infection and then migrates into the circulatory system, leading to infection of that lining of the heart. You do not necessarily have to have septicemia associated with this because it can stay contained in this area. It's usually like what we would call a focal infection. You have an infection in one area and then it spreads to this other area secondarily. But it can be caused by trauma to the vascular system and circulating immune complexes that can get caught in that tissue as well, leading to our type 3 hypersensitivity reactions. Signs and symptoms are going to be pretty much similar to having an infection in general, but you'll also have some cardiac issues as well. And then we have some very defining signs and symptoms of side pain, Janeway lesions, and Osler's nodes. These are effects that we will see apparent in patients that are presenting with these diseases. Let me see if I have any pictures. Of course I don't. My old slides used to, but I updated these from McGraw-Hill. They took out all the pictures to make it more like accessible friendly for like people with vision impairment and things like that, which is great, but now I don't have pictures. It made me sad. So I'm gonna have to fix that. But Janeway lesions and Osler's nodes, here we're talking about like spots under the nails that look like if you slam your finger in the door and you get blood under your nail and you'll see it like kind of streak out under the nail. It's like that, but without having to slam your finger in a door. You can also see this in toenails or in calloused areas of skin that you'll see these spots developing. So this can be acute or subacute and just depends on, again, how you get the disease, where it's presenting, if it's focal or if it is the primary infection and things like that. And the cause, of course, because all sorts of bacteria can lead to this. But enlarged spleen, clubbed fingers and toes are common in our subacute. So if we're talking about subacute endocarditis, something that's like slower progression, less severe symptoms, then if you have ever seen anybody that has clubbed fingers or toes, then you might know what I'm talking about. It's like if my fingers look, I don't know, I want to say normal, but I'm not trying to be mean, okay? Like the typical what you would think of fingernails. And then you see some people who have like it sort of rounded on their fingernails and it's like it rounds over, that's clubbed. And there is always a like biological reason for that. It's not a normal thing to have. So there's usually a deficiency in some sort of vitamin or an underlying infection. It's not usually just genetic. You can have predisposition for it. There's usually some other thing that's helping manifest those symptoms. All right, next is sepsis. Like we said, this is actual infection, flourishing or infection within the bloodstream itself. This can be bacteria and fungi, typically, that are going to have these effects. If you have viremia, like a viral infection in the bloodstream, this isn't anything really new. That's kind of how viruses get around in general. You can have things like cytokine storms, but it's not like expected like it is when you have true septicemia. You should not have any bacteria ever in that bloodstream. Oh my gosh, and your body will react to it. Having fever, altered mental state, high fever can accompany this. That can lead to death just from that high fever and even lead to brain damage from it. Breathing rate will increase. Respiratory alkalosis, which has to deal with you're not getting that proper gas exchange happening when you are breathing. And so you have alkalinity of the blood, pH, because it's not being managed. You're breathing in and out too quickly and not getting rid of your waste products. And then loss of blood pressure and collapse of vasculature that can lead to death. So endotoxic shock is something that can happen as a result of septicemia with, if it's endotoxin, gram negatives, right? So gram negatives, their LPS or lipopolysaccharide on their surface is also known as endotoxin. A couple things that I told you guys I would not shut up about. One of them was endotoxin, right? Being on gram negatives. We have a massive inflammatory response just from seeing endotoxin in the bloodstream, no matter what that gram negative is. And some gram positives have certain molecules that have a similar effect. We already talked about plague, so I'm not going to get into this too much, but it is hella serious. If you are lucky enough to have the bubonic version, you have the most tame version of the plague, which is quite surprising to hear about since we always think of bubonic plague as being so serious. Because it is. It's actually still pretty deadly. Septicemic plague is where you have it spreading into the bloodstream, and then pneumonic plague is when it spreads into the respiratory tract. It can be spread from person to person in the pneumonic form. If you had a blood transfusion from somebody that had septicemic plague, you could get it, but like it just doesn't really happen. I mean, if somebody's that sick, we're not going to be transfusing their blood to somebody else. But yeah, it can happen. Septicemic is the most deadly version of it. If we can treat you, you still have a 30 to 50% mortality rate, but it's 100 if you get septicemic plague and don't get treated. That is the Black Death, by the way, says septicemic version, because as we had spread of the bacteria into the bloodstream and into the extremities, they block the passage of oxygen into those tissues, and they also create toxins that lead to cellular death in those tissues, and that involves necrosis and then the blackening of the flesh, black color of the flesh. And we talked about where it's located. This is real. It still is in the United States. We haven't had any endemic cases here in Oklahoma, but we have a very healthy prairie dog population, which is where it is maintained in the Four Corners area. We also already talked about tularemia, so I don't have to go into this one too much, but we did see this is actually a more serious disease than you might think from a disease that you've probably never heard of. But it is a serious concern, and it is also a category A bioterrorism agent, just like the plague and just like anthrax. Lyme disease. So this is also cardiovascular lymphatic disease. It's caused by bacteria. It's spread by a tick bite vector. Yes, the tick is the vector, the bite spreads it. We get this characteristic bullseye rash that not everybody will present with, but it is probably more common than not. Then you can develop fever, headache, stiff neck, dizziness, and neurological symptoms after that. It's very slow to progress. And after months of being infected with this, it can lead to polyarthritis as well. The bacteria is Borrelia burgdorferi. And this is going to invade your immune system. It has a spiral shape that can kind of like at the end of them, they'll have these hooks that can hook onto the cell surfaces and things like that. It changes its surface protein so that your immune system can't find it. And it is very difficult to treat. It's also kind of difficult to diagnose. All right. And again, it's spread by ticks, maintained in animal reservoirs. Next, we have infectious mononucleosis. This is caused by a virus called Epstein-Barr virus. It is a herpes virus, so it's going to hide out into your DNA. So this virus will splice itself into your DNA and hang out there for the rest of your life once you have it. At this point, more than 90% of the world's population has been infected with Epstein-Barr virus. Probably everybody in this room at some point has had it, even if you haven't had symptoms of mono. When you are young, this says down here at the bottom, infection during the teen years results in disease about 30 to 70% of the time. So you may not have had symptoms. I didn't have symptoms when I had mono, but I have antibodies for it. So I had it at some point. I don't know, but it's there. If you've had it and even didn't have symptoms, it's still there in your body and could potentially reactivate like other herpes viruses can. I had a student a couple semesters ago who his dad would have reactivated mono, which I thought was so weird. I had never heard about that before, but that's how I learned that that's a thing, that it can actually reactivate. And of course, like we had mentioned, if your immune system becomes compromised from AIDS, the DNA splicing into your DNA can lead to presentation of cancer in the lymph nodes where this virus would hide out. It's called Burkitt's lymphoma. Next, we have anthrax. Oh my gosh, we haven't talked about this enough, but anthrax is caused by Bacillus anthracis. So it's bacterium, and because it's Bacillus, we now know that it is endospore forming. It is rod shaped, and because it's endospore forming, it has to be gram positive. So we know some things about it right off the bat. It's the endospores that really make this one a tricky one. We talked about it in the skin version, and now this is the cardiovascular. You can have a respiratory version, and you can have a GI version too. So you can just get anthrax pretty much anytime those endospores get anywhere into the body. And I don't really have much else to say about it. We've pretty much gotten to know this pretty well. So now we're going to talk about some of our hemorrhagic fevers. We've already met Ebola, and by meeting Ebola, we've kind of already met Marburg as well. They're very, very similar. They're both caused by viruses that are in the same family. So we won't really talk about either of those, but hemorrhagic fevers This is literally talking about hemorrhaging. And so we know what it is to hemorrhage, to bleed out. And that's what these viruses cause. They cause you to bleed out because having a viral infection with these guys, it really weakens your vasculature. And then your vessels will literally eventually weaken to the point that they will rupture. It starts out small and then progresses to the point that we are literally having blood coming out of our eyes and other orifices. So it can be very severe. Some of them not as bad as others. Most of these, it says all of these ones, okay? There's other ones that are hemorrhagic fevers that aren't carried by mosquitoes, but these are carried by Aedes mosquitoes. We have Aedes aegypti here in the United States, so we can potentially house some of these hemorrhagic fevers. And guess what? Some of these we do. And most people don't know that either. So obviously we don't have yellow fever. This is more found in South America, and you can have vaccination for this, okay? Dengue fever. Mostly we found this in South and Central America, the Caribbean, Asia, and Africa, basically any tropical area that had these mosquitoes, but they have moved up into the United States. We do have dengue present in Florida and parts of Texas. So it has started to creep into the United States, almost endemic at this point. It's pretty uncommon to get a case of it, but those mosquitoes are moving further and further north. And so are the viruses and other diseases that they carry. So we need to be aware of that. Chikungunya might sound like a strange disease to you and I. It's a pretty tame one as far as hemorrhagic fevers go. It doesn't always present with hemorrhagic fever, but it is also in the United States and actually become endemic here. We have it here in like very southern portions of the United States. And we'll see a few cases here and there in Oklahoma as well. It's not super common. And again, it doesn't always have like hemorrhagic fever associated with it. Most people will get over that one just fine. Not all of these are like 90% mortality rate, okay? Not like Ebola and Marburg, that we have certain strains of these that are like at the low end with the 50% mortality, and then at most of them at the high end with the 90% mortality, depending on what strain we're dealing with. Ebola in general, the ones that we have present here in humans on here, mostly in Africa, is where it's endemic. It is, we think the reservoir is in bats, so we haven't been able to prove that yet. And then it'll get spread to primates, and that's usually going to spread to people by people in villages who are hunting for bushmeat. And that's how they butcher their meat for them to eat and their food, and then the blood spreads it into the human population. It's pretty limiting in the human population because for the most part, people see people who are bleeding out and they don't want to engage with that. But so outbreaks don't tend to become like pandemic in nature unless we have travel across continents and then a person is sick, and then usually it doesn't get too far in the other countries, but it is an extremely deadly disease and you do not have a good prognosis if you do have symptoms with it, even in good healthcare facilities. There is an airborne version of Ebola, but it's not in humans right now. It's only found in primates. We found this by accident. We ship in primates from the wild for use in like medical research and things like that. And the Army shipped in some primates from Africa and put them in quarantine like you do to make sure they didn't have any other diseases. You want to be sure that what you're working on doesn't already have a disease. And it turned out that one of them had an airborne version of Ebola. And that was here that they discovered that here in the United States. And it was a monkey that they believed they found, the source monkey, that had been kept in a completely different room from other quarantined monkeys. And those monkeys also got Ebola. And that's how they found out that it was airborne. They had to quarantine everybody who was working in the building at the time, and nobody developed that airborne version of Ebola. And that version of Ebola is called Ebola Reston, for Reston, Virginia, where this was discovered. So it's terrifying to think there are strains of Ebola out there that are airborne. I don't want to think about like the likelihood of that being used as a bioterrorist weapon. Then we have Lassa fever. This one is strange. So most cases are going to be asymptomatic. It's like you either 80% of people don't have any symptoms, and then 20% of people that have the symptoms, you're going to have hemorrhagic fever, and it's extremely severe. So it's a strange one. But again, another hemorrhagic fever. And let's go on. So I'm going to say about it. Okay, so let's move on to the non-hemorrhagic fevers. These are going to be fevers, so they actually have a true fever associated with it. You're going to have a fever. I mean, a lot of diseases do, but we're talking about a circulatory and lymphatic disease associated with fever. It's present in the bloodstream. A lot of these are tick-borne. Okay, so some of these are bacterial and some of these are going to be protozoa. Brucellosis is a bacterium transmitted by tick bite, and I don't know, it's mostly maintained in ungulates, so we're talking about cattle and things like that. Oh, sorry, no, I'm not. That's ungulate. I'm like looking at ungulant fever, I'm thinking ungulant fever, and that's a different disease. So brucellosis, tick bite, ungulant fever, because the temperature goes up and down throughout the whole disease. We don't really know the mechanics of why that happens, but it does. That's why undulating temperature, that's what that's talking about there. And the phagocytic cells have a good part to play in how the disease presents itself, but there's no hemorrhaging, it's non-hemorrhagic. Q fever, this is the one I was thinking of with the cattle. So this stands for query because they didn't understand what was causing the disease. It's caused by Coxiella burnetii, maintained in the animal population, transmitted by ticks. Most of these can pretty much be summed up as tick bite diseases, and I'm probably not going to ask you a whole lot about a lot of them. However, Bartonella henselae is not necessarily tick-borne, okay? You can get this from cat scratches, literally, which is why it's called cat scratch disease. Even as you might think that this is something that little kids only get, but I had a guy, again, a couple semesters ago who was in the Air Force, very fit and everything. He got a cat and his cat scratched him and he developed so severe of cat scratch disease, he was in the hospital for over a month getting treatment for this. It spreads into your lymphatic system and you get these buboes. And anytime you have an actual infected lymph node, not just the inflammatory response building to other areas of infection, but the actual infection residing in a lymph node is called a bubo when it swells up like that. So we have these lymph nodes that are going to be actually filled with pus and be infected. We have high fever. About 40% of cats will carry this just naturally and usually get it from a bite or a scratch. Next, we have trench fever. This is also Bartonella, but this one is Bartonella quintana. This is liced. So this is very common in homeless populations, especially in San Francisco, where it is a huge problem, the homeless population is. And they spread the lice amongst one another, and therefore they spread the disease amongst one another. So about 33% of homeless folks in San Francisco have this bacterial disease. You get sort of feeling sick symptoms, and then you can develop endocarditis as a result as well. Next is ehrlichiosis and anaplasmosis. So these guys, Ehrlichia, Chafensis, and Anaplasma phagocytophilium, these are tick-borne diseases. We have ehrlichiosis in the United States. We have both of these in the United States, but ehrlichiosis will actually test this and ask you questions about it. And if you donate blood so that we don't transmit this in transfusions, but you feel sick, you'll have some muscle pain, rigors, so stiffening of the muscles. And you usually are going to recover from this just fine without any effects as long as you have a healthy immune system. That's how most of these are, if I'm being honest, okay? Babesiosis, this one's protozoan, okay? So similar to Ehrlichia and Anaplasma, but the protozoa version of it. Next, we have Rocky Mountain spotted fever. Don't confuse this one with Lyme disease, okay? They're both Lyme disease and spotted fever. These guys are both bacterial tick-borne infections, but you guys have to actually know the organisms, know the difference, okay? So Lyme disease, we had that slow progression associated with Borrelia burgdorferi. We had the weird neurological, the bullseye rash, and the arthritis. But Rocky Mountain spotted fever is borderline potentially hemorrhagic in a way. We call it non-hemorrhagic because that's how it presents in most cases. But in a lot of cases, it totally play by the rules. You'll get feeling like you're sick. You'll get some spotted rash. It's usually petechial in nature. So we have little tiny burst capillaries under the skin. And that happens about 2 to 4 days after the prodrome. We haven't seen this term in a while. So I'll remind you guys what a prodrome is. Prodrome is when you see your very first symptoms. So when you start very first feeling, kind of don't feel good, that's the prodrome. So when you first start having your very first symptoms, 2 to 4 days later, you get this rash. It's petechial rash. The rash lesions can merge eventually and become necrotic. So this can become pretty severe and lead to necrosis of tissue, cardiovascular disruption, hypotension, thrombosis, hemorrhage, shock even. It has a 20% mortality if you don't treat it. And even if you do treat a student of mine, I don't know how many freaking tick-borne diseases you can have and whatever, but and another student of mine, separate from these others, that she was a paramedic or EMT and went to a rural location and got a tick bite and ended up getting the Rocky Mountain spotted fever, and she was hospitalized for it for also over a month to get treated for this. It's serious. When we think of just being a tick-borne disease, oh, you might get Rocky Mountain spotted fever. Oh, you might get Lyme disease. But these are actually really severe diseases that you do not want to have. And it's not like you're like, oh, I think I had that maybe. No, you don't kind of think you might have had that. So this is very indicative of that rash that it would be associated with having these. Now, a lot of these tick-borne diseases are associated with certain kinds of ticks, whether it's the dog ticks or the exotic ticks. So exotic are hard, and they're usually like the deer ticks, the little ones that are like crunchy. Then you have the ones that swell up all gross, and those are like the dog ticks that we typically think of. So they're different kinds of ticks, and they can carry different kinds of diseases than one another. Okay. Chagas disease, this is caused by Trypanosoma cruzi. So that's our protozoa. This is an assassin bug or kissing bug. These guys will bite you with like actual bite you. There's not like a poking in like how we have with mosquitoes and with a proboscis. These guys kind of bite into the skin, lap up the blood, and then poop out parasites that they carry in their body. And then those migrate into the skin or the wound. And then inflammation develops from that they call a chagoma. And this is Chagas disease. That's the first sign that it's happening. You'll have a mild fever and feeling just sick in general. And then eventually it can become chronic version where you have inflammation of the heart, disruption of the heart's ability to function, the brain, the intestinal tract, and it can be deadly. This is an issue. And you might be looking at this and like, wow, that's crazy. Like, glad we don't have that. You do. We do have that here in Oklahoma. If you guys have ever donated blood, that's one of the things they'll ask you about. And they'll actually test every single person that ever donates blood for Chagas disease. So you should get notified about it. Yes.
I remember a doctor told me it was only in South America. Also, I don't think assassin bugs do it. I think it's just kissing bugs, as far as I'm aware.
Kissing bugs are assassin bugs.
Well, assassin bugs are not.
Not all assassin bugs are kissing bugs, but
Yeah,
kissing bugs are all assassin bugs.
Something like that. Those
So-
are the great ones, though.
With the wheel on the back,
I love those ones.
those are wheel bugs. Yeah, they're related, but they're not. Those don't.
Never spread a disease.
No. They're good for your garden. By the way, the wheel bugs are really good for your garden. You should have them. But these guys are not. And you will see these in your house. I've caught several of these on several of the places that I've lived here in Oklahoma. Not all of them have Chagas, right? But like they have the potential to carry it. And there are bugs in Oklahoma that do have this. It's a lot more common in like Texas and further south. But it is here in the United States. It is endemic. So they do, you do want to keep an eye out for these. They nest in warm areas, like to live in your house. So keep an eye out for them, especially during the winter time. Spray for bugs and yeah. They will mostly attack children because children will sleep a little bit more soundly when they are asleep. And will like crawl on them and then bite them near the face, the eyes. They're kind of attracted to the carbon dioxide. And it's very common to get inflammation, the chagoma near the eye area. We've seen trypanosoma before. The one we saw was the African sleeping sickness one in our slides, but you remember the little squirrely guy that I had you guys make sure that you saw. This is a field of red blood cells, and that little squirrely, squiggly guy, that's the trypanosome. All right, malaria is our next one. This is a protozoal infection. These protozoa get into your bloodstream from a mosquito bite that regurgitates its meal into you. And then you have this protozoa in your bloodstream. It lives in your actual blood vessels as part of its life cycle. Not blood vessels, red cells. That's what I'm trying to say. They live in the red cells and they'll like burst out periodically with their little larvae to go into the next part of their life cycle. They can also live very easily in your liver and in your spleen. So if you've had malaria, you may never donate blood because they don't want to risk that recurrence happening because it can hide out and become latent in your tissues. So be very serious. So usually have cycling of symptoms over 48 to 72 hour periods, fever and aches and pains. And that's usually when we have reemergence of those organisms from like the blood cells, like them bursting out and going back through their life cycle. The worst form is the falciparum version. And that's because these organisms are named Plasmodium something. And the most common and worst one to have is Plasmodium falciparum. That's the name of the organism. But it can lead to death and coma, and usually associated with damage caused by blood vessels as it spreads throughout the body if it's left untreated. We treat this with quinine and related drugs, and we also have other drugs that are available. There are vaccines available, but they are not going to be effective if we don't get boosters. And where this is the most concern will be in rural areas of sub-Saharan Africa where people aren't going to get boosters. So it's hard to control that. So most of the time when we're controlling malaria, we're controlling the mosquito population. So we want people to have mosquito nets. We want to have mosquito abatement efforts to kill the mosquitoes in those areas. So pesticides and things like that to help control that. You also, if you're traveling to areas that are endemic for malaria, you want to take anti-malarial drugs before you go there to get it in your bloodstream to have it disseminated to protect you. So here's the picture of the life cycle. I'm not going to go through it. I will say in malaria, the mosquitoes themselves are actually infected in their gut. Their gut cells are infected with the malaria. It's part of their life cycle. So they're not just carrying it around. They are actual biological vectors, not just mechanical ones. They are infected with it. Also, the parasite can undergo antigenic variation, meaning that it can shift its surface protein. You think you've got antibodies against it? Nope, it'll just change it up and that'll help it survive. So yes, we already talked about all this. Oh, they're also introducing sterile male mosquitoes into populations that were genetically manipulated so that they are sterile. Mate with the females. The females can only mate once and then it kills the females. So it helps control the population that way. HIV is caused by a retrovirus. It's called a retrovirus because we have that reverse transcriptase. So its genome is RNA, turns it into DNA, and then incorporates it into the host genome, thanks to that enzyme and other accompanying enzymes. We have a lot of association, or I would say misunderstanding between HIV and AIDS. AIDS is a syndrome. It's acquired immunodeficiency syndrome. So it's a collection of symptoms. And usually you aren't going to develop AIDS unless you have had HIV, but you can have HIV and not have AIDS. So as long as you have the HIV virus in you, you are HIV positive, but that doesn't mean you have AIDS. AIDS is something that happens when you get those symptoms. And that's usually associated with a very low T cell count. So if we can keep your viral load close to 0, we can actually prevent you from having AIDS. But close to 0 just means it's undetectable. Did I tell you guys about the person that tried to, okay, there's somebody on Instagram or something, some podcast or something who was HIV positive and he had a new partner and he hadn't disclosed that he was HIV positive and then had to having a discussion about it for some reason. I don't know, they were getting in an actual relationship. And he was like, yeah, you know, I'm undetectable, but I have HIV. And he was like, well, I don't know if I want to be in this relationship. Like that's dangerous. I don't know if I'm ready to risk my health and well-being for this. And the person who was infected got mad because he says, I'm undetectable though. Like I shouldn't have to tell you. I mean, basically like, it's like, if I can't get you sick, why does it matter? But just because you are undetectable does not mean you cannot infect people. It means the machines can't detect the viruses. It means the blood sample that you gave that day on the test said there weren't any viruses. It does not mean you don't ever make a virus and can't ever infect somebody. So irresponsible. And their whole argument on that whole podcast was that why should you have to tell somebody that? And the people on the podcast were in agreement with that. Appalling. Appalling. Imagine that you get into a relationship with somebody that is HIV positive, but is undetectable. And then you end up with HIV and they're just like, well, I'm undetectable. That wasn't for me. You must have been cheating on me or something. Oh my God. So, right, that's what I say. Hell no. So advocate, those of you who are out there, keep reminding your patients when you get into the healthcare field, undetectable does not mean that you aren't contagious, okay? It just means you're undetectable, that's all it means. But there's all sorts of things that you can develop as opportunistic associated with AIDS. So that's a sign that your T cells are declining and your immune system isn't keeping things in check, like these types of cancers and diseases that are really only associated with having a poor immune function. And again, AIDS is defined as having this really low T cell count and an increase in opportunistic infections, as well as an increase in the viral antigen in the blood as well. We kind of got over this, but you're going to have wasting, you're going to have symptoms of general diseases and presentation of extremely unlikely diseases as well. So retroviruses belong to the lentivirus family. There's 2 kinds of HIV, but HIV-1 is the most common. Reverse transcriptase turns RNA into DNA. This kind of talks about how that whole process would work, how it hides out in the DNA, and it can make more virus later on as it sees fit. It's transmitted from basically trauma exposure to blood, needle sharing, sexual transmission. They say it's related more to micro tears than it is necessarily the direct fluid swapping, and then breast milk. But we don't have transmission through urine, tears, sweat, or saliva. And I'm not going to talk too much about epidemiology. We know it appeared in the 80s. They called it a gay-related disease, and it is not. We use ELISA, certain agglutination with antibodies, and other rapid antibody tests to diagnose this. In order to have AIDS, you have to have HIV and a low T cell count. You want to prevent this by avoiding sexual contact with people that have HIV, using barrier protection if they do. And we also have PrEP and things like that available now, where you can take antiretrovirals and have relationships with people who might be HIV positive to protect yourself from getting the infection. Don't share needles, or if you're gonna share needles, bleach them in between. It's on the slide, okay? So if you're gonna use your needles and share them, please bleach them before you share them. Yeah, there are vaccines that are undergoing trial, but there haven't been any promising ones so far. We want to inhibit all sorts of these unique factors of HIV, including the reverse transcriptase. The integrase that it makes is a specific protein that it uses to put its DNA into your DNA. And it has a protease, which is an enzyme that's it makes its whole proteins, like all of them, all in one long translation, and then clips them into like the usable proteins. That enzyme that clips them is called protease, and we can inhibit that because all of those are not things that we have. Might make it seem like it's an easy thing to prevent, but obviously it's not. It mutates very easily. That's that. Okay, so let's have I would say mini break. I'm gonna pull up the Kahoot. Y'all chill. We're gonna shift into lab mode. We're not even gonna go up to lab because that place is nasty. So, and then yeah, if you guys from online, like Jonathan, if you guys wanted to stay for this one, I'm not taking attendance for the online one, so just save you the trouble from having to. Yeah. All righty. I want this to go away. That's what I want. There it goes. There it is. Everybody was here today, right? Except for Eileen. I thought so. That's right. So I know Renee's on. Anyway, she's so there, yeah. Yeah. I did it. I still have that question. Oh, I don't imagine it. I was like, it's fine. Yeah.
bring them to one. I think I have to please let me know. Like, why would you not follow up on that email? It was quite it. Anyway. I read it like all the oh my god. Right, but I literally had a full like panic attack. I was like, yeah, I was falling. I would start to shower. There was like
I'm just vaguely overhearing this, like you're talking about this test, like it was a test you didn't know that you were supposed to take.
no, so for
Right.
your test and your background check, and I did it like on time, like weeks and Sweeney, she sent me an email, but every single time I would look up her name when I was looking rolling, and I would me an email saying, Sarah, I haven't received either of these from you yet. It was October 22nd, which is I saw it last night, and I was like, it was due October 21st.
You're like, I got this taken care of.
I literally have the receipt. But I was like, I'm so sorry, I'm just I have the receipt. I will come bring them to you tomorrow.
Right.
Let me know.
Right.
But I have like a full on panic attack last night.
Yeah, I'm that person too. Like, even though you like, part of your brain knows that you're freaking out over nothing because you know you took care of it, but then the other part is like, what if they're assholes? Like,
My thing is, what if they don't give you the results either, so you don't know like when it's gone like through or anything.
Yeah. Right.
Because it goes directly to the
Right. Yeah.
And so like, how am I supposed to know that?
Right.
I still have to think about it. Yeah, no, they don't tell you.
Yeah.
They fail.
The job that I had before this did drug testing like that, where it's like I couldn't pee either, and I drank water before I went and drank water. I guess because the nerves or something, like my body just wasn't having it.
Same. One time I did actually have a mental breakdown because I couldn't get a pee sample, and then
They were getting mad at me too. They were like, if you can't pee in the next hour, you're not going to be able to do this test. I'm like, I have drank so much water. You guys have no idea. Like, I want to pee for you. I genuinely want to. Finally, I got a little enough out to do the test, but it was just like one of those things where it ended up being like, oh my God, I'm not going to get this job because I can't pee right now.
that's so dumb. And like freaking out over them freaking out over pee.
Yeah. Yeah.
That's it. All the money already.
Did they get back to you and tell you to be good? Okay. Oh God.
I haven't emailed her back saying.
We'll go ahead and get started with our lab review. This probably won't take too long. Remember, these are like going to be like a question from one thing. We're not going over every single detail of every single thing. I said we'd have a review. Did I say we would get 8?
Yeah.
Okay, I'll have a review at 3 p.m. Probably, I hope that I can get home and do it from home because I just don't want to sit at this building. So, so we'll try. I do need to do some cultures for the exam too. So it just depends on what I get done with, but it's not you guys' problem. Huh?
So I'm fine with fine.
I don't really have a problem with that. I just don't want to like stretch out because my husband's like off today. So I always feel bad when I do work when he's home. He just, believe me, he does enough to make me be aware that he's not so keen on it either. But anyways, after all the things that have happened, being on faculty senate this semester, after all the things that I have noticed about how this place doesn't work the way that I feel like it should work, and how like inefficient, because you guys know, like I'm really on top of my shit and I'm an overachiever. And and I don't feel like people stand up to like what I expect of them as faculty. With students is a different thing because like I'm there to help you figure that out. But with faculty, I expect them to rise to a certain bar, and I hear about some of my students complaining about certain adjunct professors like not appearing in class on time, or
Professor Bird.
Yeah. Right. So there's no like evaluation system in place for adjunct. So I am the one writing that policy. But anyways, having done all, I am applying for a summer start at a university. Doesn't matter, it's Bradley University, but it's for an online doctorate in education for admin track. So it's a 2-year program to get a doctorate, so I can become an administrator at this university and make bigger changes. So, yeah, so I've requested, I already have really good support from all sorts of people. So I've got, recommendation letters from my dean and from Dr. Nephew, her, she's the president of faculty senate, and she's great. I mean, if anybody does work, she does. Like, she really cares about her students and making this place work right. So I got recommendation letter for her from Tiffany Smith and our interim dean, and probably from Dr. Rhodes too. He is, I don't know if you guys know this, but he's not a morning person. So I emailed him this morning and he's the one that hasn't replied, but I'm not concerned about that. It's just, they'll be mine, and then I'll start in the summer, and hopefully if I see you guys around, you can ask me about how my classes are going. And because I'll still be working full time with all of that too. So
Being a doctor and working here.
yeah, yes. Yeah, it's online, so I don't know. You guys know me. I'm already doing work 24/7 anyway, so might as well like be getting a doctorate while I'm doing.
Why not? True.
Yeah.
That's true.
So then I can get paid more and make better changes for you guys when you're at nursing school and future yous going through. So let's get started with this. Yay! I'm excited about it too. Okay. First, I have to get accepted to the program, so I mean. If I don't, then I don't know. That must be very prestigious, I guess. I don't know. All right. So we're going to have 24 to 26 stations. It's usually going to be 25. And we'll have the 50 questions, multiple choice. It's not 100 for the lab. Okay. It's just 50 for the lab. And 1 minute per station again, Scantron again, all the same as last time, basically, just different topics. All right, let's talk about our culturing basics. We already did this stuff, so this should be definitely review for people on that stuff. So we'll go over some of this stuff really quickly. What is most commonly used to inoculate cultures? An easy one, just to get us in the mood.
Definitely.
Yes. Your tone. All right. So obvious one there, the loop. I would hope you would be able to answer that correctly, even if you hadn't taken this class. All right, in which quadrant of a streak plate would you expect to find the most single isolated colonies to start new cultures, perhaps pure cultures? All right, and you guys saw this question on the test last time. Pretty much the same question. It might be a little bit different, but it's almost always the same diagram and everything. You'll have it again on this test as well. All right, so remember your aseptic technique, why we're doing it, how to do it properly, your lab safety, the types of media. Here I'm talking about broth versus slant versus plate versus butt, those types of media, okay? And which ones are solid, what auger is, that stuff. Different ways to inoculate the media, whether we're stabbing it, using the loop, swishing it with the loop, and that sort of stuff. Because that is another thing that I will ask you about on the exam, which is like, how is this media inoculated? So if I give you that orange slant that we know that we did the streak and the stab on, you'll have to know that we did both. Most of them, when I get to asking that question on them, they'll all have the same choices. So there's not going to be like giveaway choices on those types of things. Okay, so I'll have like stab, streak the slant, swish in the broth, or streak and stab, that sort of stuff. And you'll have to know that for all of the media, which is all on the study sheet, by the way. All right, stains. Let's review some of these guys. What's your favorite? What is the first step in the gram stain procedure? All right, so most of us got this one. This is crystal violet. You're going to put crystal violet. Well, you're going to heat fix a smear first. Yeah. Then that was one of the choices. So our first step for the staining process is crystal violet. Then rinse. Then iodine to fix the crystal violet in place. Then rinse. Then alcohol to rinse out any crystal violet that didn't stick. And then rinse with water. And then you're going to saffron in for the counter stain to help you see the ones that did rinse out. Now look at what you guys have learned. You actually learned a thing. Good job. We know gram stains at least. Okay, so what color do properly stained gram-negative bacteria stain in gram stain procedure? Some of these pictures are just pictures. If I think they're important, I'll tell you guys. This is just a picture. Sorry. All right. Negative is going to be red. That's our negative color. Purple is positive, right? You can think of red as being pink if you like to because it looks pink, but red definitely doesn't look like purple, right? So keep that in mind. Purple is positive. What is the name of this staining procedure? Uh-oh. Excellent. So this one's the endospore stain. So we have the red cells and those greenish bluish structures are the endospores themselves. Some of the endospores look like little bubbles. Some of the endospores just look like green spots in the red. So we might have one or the other or both in the slide that you will see. You will have an endospore stain. And it'll be better one than we used the last one. Which of the following bacteria will be acid-fast positive based on their names? And this is an image of an acid-fast stain that is positive. Okay, so this is Mycobacterium leae. At this point, remember, we kind of were pretty good at determining that it should have been myco something. Mycobacterium, but mycoplasma, those are the guys without the cell wall. And the mycoplasma, I always think plasma, like I said, if you had a cup of plasma and spill it, it doesn't have a shape because it doesn't have a cell wall. Okay, that's what's important about those guys. The mycobacterium have the wax on the surface, the mycolic acid, binds with the carbofuchsin, it's lipid soluble, and it stains it that fuchsia pink color. And our counter stain for that is the methylene blue. So our negative cells will be blue. And the same slide that you guys saw the last time with that mixture of both the blue and the pink is the same slide that you'll see again. Okay. Let me know. I only have so many slides. I'm going to be transparent here. All right. After the slide has been heat fixed, when do we use heat again in the acid fast stain procedure? Okay. Concerning. So this one, acid fast, is a little bit of a trick question. We do not use heat in the acid fast procedure. Do you guys remember what procedure we use heat on? The endospore stain, right? So that's the one with that steam and the nasty green drops and everything like that. So that's endospore stain. We do not use heat in our acid fast stain, okay? Also, so the 10 minutes over steam, endospore. Drying a stain into the slide faster, that is not something we ever did as far as trying to dry a stain onto something faster with a Bunsen burner. Flaming it again to fix the primary stain. I don't think we ever flamed slides after putting a stain on, like directly over the flame. So none of those should have applied. The yellow and the green shouldn't apply. So again, don't freak yourselves out. Actually read and understand your choices here first. What color is a negative acid fast stain? At least on the exam, you don't really have a timer. Like we have the 1 minute, but you can always go back to anything. So don't rush yourselves on the actual exam. Negative acid fast. So I did just say this one, so hopefully you guys did hear me say it is going to be blue. Our methylene blue is our counter stain for that. So our negatives are usually our counter stains. What is the name of the primary stain used in the capsule staining procedure? The first stain we use. This is a picture of a capsule stain. Does this organism have capsules? Based on this picture? Yeah. The white around those pink cells is the capsule. If you don't have a capsule, there will be no white at all. So the name of the primary stain that very first stain we use is Congo red, or the capsule stain. You put it in with that Congo red and we smeared it out, let it dry. And then what did we put on it after that? Do you guys remember what that was? Manivals. You turn it that purple color. All right, in which staining procedure do we use the stain? So I already said this one. Let's see if you guys remember. My eye is like weeping. All right, that's the endospore stain. We're gonna force the malachite green into the endospore itself, that cuticle. So for our stains, same as last time. You do need to know the procedure. So you need to know as much information about the stains as you had to know for the midterm for the final. I'm not watering that down, okay? So you do actually have to know all of that stuff. Just say it that way. You guys know what to expect now. So differential and selective media. Let's get into this. So what type of media is this? This is like a common question you're gonna get. I'm gonna give you a plate or something and you're gonna have to tell me what media it is. So you should be able to tell me what media this is just by looking at this picture. Right, so this one is EMB, eosin methylene blue, which is our selective agent in this as well, because it's selective and differential. So it's selective because we have the EMB that inhibits gram-positives, and it's differential because we can tell for lactose usage. If it uses lactose, it turns pink. If it aggressively uses that lactose, it turns metallic green. So that was showing that metallic green. This is the only media that does that, so that metallic green was the giveaway. If the dark color didn't give it away for you. All right, what is the selective agent in this media? Same one, I just said it because you guys remember when I said the selective agent was. All right, again, this is very similar to the questions that you're going to get on the test. So that's EMB, eosin methylene blue, prohibits gram-positive growth. What media is this one? Looks like we have a pinkish reddish agar with some pink growth on it. All right, so this one is McConkey agar. EMB and McConkey are the ones with lactose use tests on them. And if you use lactose, you turn pink. The growth itself will turn pink, not necessarily the plate. The plate just happens to be a pinkish reddish color. Remember McConkey agar, our selective agent is biosalts. The lactose use is our differential. Is this media selective or differential? Uh-oh, I did just say, let's see if you caught it. You can choose more than one. All right, good job, guys. You were listening to it's both differential and selective. All right, which media is this one? I feel like this one's a pretty easy one, but I don't want to get ahead of myself. All right, we have mannitol salts agar. It starts out that reddish color, that coral red color, and then it can change colors based on if something grows on it or not. Our selective agent is just salt proper. And so our gram positives will grow on it more easily than the gram negatives. And then our differential is our null red pH indicator. It will turn yellow if you are able to ferment the mannitol in it. And it will turn pink if you are just breaking down like the protein and nitrogen source in it. And it might not change any color at all, and it might not grow at all. Favors gram positives like Staph aureus. What is the pH indicator used in this medium? Probably the same thing. Yeah, it is. Again, another very common question on this, and I'll probably give you every pH indicator that I have. Phenol red, litmus, bromothymol blue, and I might even throw in a bromophenol blue. So be sure you know your terms. All right, so that's phenol red. If it starts looking red and turns pink or yellow, it is phenol red. If it starts lavender and it turns pink or blue, it is litmus. If it starts green and it might turn blue, then it is bromothymol blue. That's all three of them. They are not at all like each other. All right, so our differential and selective media have some aspect of our other media that we're going to get into as well. But know what makes them selective, know what makes them differential. If I give you blood agar, it is not selective, it is enriched, right? But is it differential? Yeah, because we can tell if we have what kind of hemolysis it is. Know the different kinds of hemolysis. The alpha being the greening, the beta being the total clearing, and the gamma being just it grew on it and didn't do anything else. Now let's move into the tubes, shall we? Yay. So most of the test will be the tubes. You guys already know that though. Which test is this one? This is very much like what you will see on the exam. I'll have actual tubes though, and you are allowed to pick them up. All right, this is citrate. All right, so citrate and malonate are the ones that start out green and can turn blue. They're testing for whether or not that organism can use that molecule as the sole carbon source. If it does, it makes an alkaline byproduct, turning the bromothymol blue a blue color. So which of those tubes was positive? This is a citrate test. Which one tested positive for citrate usage? All right, excellent. So that's B. Think blue for base. Base is alkaline. Okay. And if it's turning it alkaline, it did something. That means it's positive. If it didn't, then it's not. It usually won't grow if it's negative. What is happening to the pH in tube B? Come in full circle, y'all. What I tell you about pH in chapter 2. We're going to come back to it. All right, so pH went up whenever, so blue means base for bromothymol blue. Blue means base. Base is at the high end of the pH scale. Acid is at the low end. And if you need to draw that out or just make a note of that on your scratch paper, whatever helps you remember it, you will be asked about pHs. All right, I won't maybe ask you exact numbers because I don't care. Simmons citrate is what that media is called. Okay, Simmons citrate. How do we inoculate it? It's a slant, it's just a regular slant. Okay, how do we read it? We're looking for the blue color change or no color change. Starts off green. What colors are produced? Started as green, might turn blue if it's positive. What do the colors mean? Blue means that it made an alkaline product from breaking down citrate. And what is the pH indicator? Bromothymol blue. Bromothymol blue, not phenol blue. All right, what test is this one? Oh, we haven't, oh yeah, okay, yep. I forgot that I had 6 choices on these. Your exam will have up to 5 choices. All right, so with this one, I'm trying to make this go away, sorry. With this one, this was our phenol red carbohydrate fermentation test. Fairly widespread across all of these. Nobody was really sure about this one. This is where making some flashcards with the actual color images that you guys have on the study sheets that are available. Do that because I will absolutely just put a tube in front of you and say, what is it? And I might not give you one that's inoculated to compare it to. I might just give you a tube and say, what is it? What's the pH indicator? Something like that. So what would it look like if it was positive? That sort of stuff. So this is phenol red carbohydrate fermentation. We know it's phenol red carbohydrate fermentation. Well, we know it's phenol red because it's that orangey coral color, phenol red, right? So that's our carbohydrate fermentation test because that's the one that was that coral color with the Durham tube that was upside down in it. So that's how we know how to recognize that one. We'll see a lot of the other ones. I'm not going to describe them all right now. What is the name of the upside down tube inside the assay tube? I did say it. I always forget.
That's not a.
All right, so that's a Durham tube. Good job. What does it do? It collects the gas fermentation process.
Here she is.
Trying. All righty. What are the two products of fermentation that we can detect with this assay? The two products of fermentation. Right, so pretty good job here. Gas for sure. That's why we had that Durham tube, right? That's a giveaway. Acid. So sugar is not a product of fermentation, right? That's what our starter is. Our H2S is not part of fermentation. It's part of a different process of anaerobic respiration. Okay? Different thing. And this assay doesn't test for it. So acid and gas. Which test is this one? This is all one test, just different kinds of results. All righty, so that was SIM. When you got to know, right? You got to be able to recognize these guys when they're uninoculated and what their different possible results could be. What does the I stand for in SIM? All right, so the I stands for indole. It's a product of breaking down tryptophan. If your organism can break down tryptophan, then it will produce indole. How do we detect that indole in the SIM media? Okay, another common question from the exam. Okay, so that's Kovacs reagent for that one. Again, these are like flashcard types of things. Just have to know them. Okay, how do you detect H2S, the sulfur part of the SIM? How do you detect sulfur reduction? All right, so we're going to observe that black precipitate that was very obvious in that last tube. What you might not have noticed in that last tube in that example image is that there was a layer of Kovacs reagent at the top of it. It was gold. So was that in that picture with the black and that gold layer of Kovacs reagent, is that positive for indole use or is it negative?
Negative.
Negative, exactly. What does it look like if it's positive for indole use?
Red.
Red. Very good. All right, how is motility detected in SIM? Okay, so for motility, we're going to look for growth away from the stab line. Very, very clear stab line with the growth in the stab line if it's non-modal, right? We all saw that, it was very defined. Anything deviating from that typically is going to be modal. And I will try to make it very obvious for the test. All right, how do we inoculate SIM? You guys remember? It's a butt, so we stab it. Interpretation of the results, the changes, we pretty much went over all of those, so I think we're good with that. And then addition of the reagents, remember that's COVAX. All right, which test is this one? Put up my Christmas tree last night. I'm really excited. We need to get a couple more ornaments. It feels like bland right now. It's all like, I don't even know. I put a little picture, I have a picture of it.
Oh.
But we have like a little like hemp, like a jute like ribbon garland around it, and like some little like bohemian tassel guys and some bells and whatever, pine cones. But I think we need some silver and gold ornaments too, just to make it sparkly or something. I don't know. It's pretty. It just needs a little more zhuzh. It's our first Christmas tree. I know. All right, in litmus milk, what color is associated with an increase in pH? This is litmus milk, right? We know this because litmus makes it this lavender color. But when we have an increase in pH, what color is it? So yet again, blue is base, and base is an increase in pH. Happens for the litmus milk too, not just the bromothymol blue. So anytime that you see a blue shift in color in our pH indicators, whatever they are, it's going to be alkaline, which is the same thing as base. Increase in pH, right?
Like the number in the pen to get in. Oh, never mind.
Yeah, down at the bottom. Sorry, you like asked me, and I'm like, I know it's somewhere. I'll give you a second because it'll disappear if I click next. Do you have it? It should pop up, hopefully. Some point it disappears. Here it goes. In litmus milk, white. What does it mean? Okay, so white means reduction. We're literally reducing, gain electron reduce, the litmus itself. So our pH indicator no longer has those colors. So it's not going to be lavender or pink or blue. So if you reduce it, that goes away completely. And then we just left with the color of milk, which is white. So reduction. All right, so there's a ton of colors going on, a ton of like different things going on with the litmus milk, right? So let's go through them just really quick. Original is lavender. So we start with lavender in the middle. I'm going to kind of make a little bit of a drawing on the board. So if this is your original, where you start, and it is lavender, I'll put L. Okay. For acid production, and for, actually, you know what? Let's do pH lower. So let's put it on the other side, just to keep things consistent. For acid versus alkaline. There's a couple of stops involved, okay? All right, so we know it's lavender if it's neutral or uninoculated, right? Okay, if we go to the acid direction, the first stop is pink. Pink, okay? Pink, you're making acid from fermenting the lactose, right? To make even more acid, you will reduce this and it will go white. Okay. So it's just like a scale, depending on how much acid, like how really into doing the fermenting of the lactose you are. Either of these can coagulate. So that's the solidification. Okay, chunky or solid. All right. So neutral, going back to that, if our pH is increasing, our first stop is gonna be blue. Blue, alkaline, why is that happening? We're breaking down the proteins in the milk, the casein, and making alkaline byproducts. We're not doing any fermentation though. Okay, just breaking down the proteins, we're living in there, so we can break them down. If we're really dedicated to breaking down that protein, then we will go to transparency or clearing up, okay? If you're going transparent, we could call this, the formal term for this one is digestion. So I'll put a D here actually. For digestion, it's transparent though, okay? That means you're completely digesting those proteins. So now all that's left is the liquid, like the water part and like the byproducts of them. So you'll have where you can actually see through it. Now there might still be like see through where it's like plasma colored and then a layer of dark purple at the top. The litmus is still there. It has not been reduced, right? We haven't gone that route. The litmus is still there, but it'll just kind of separate out and sit on top, okay? But if you can see through it at all, if it's gone transparent at all, that's digestion. You can, excuse me, you can sometimes have gas. This can be and or. We're not going to see that in the test. I'm just telling you, it won't be in the test. All right. What is happening with digestion? We're actually breaking down the proteins in the milk completely. How is gas detected? If we were detecting it, it would be bubbles in the agar. You can actually see air pockets, or they would be like cracked up and separated out in the coagulation. All right, and what is the sugar in milk? Lactose, right? Like lactose intolerance, not a secret there. All right, which test is this one? Okay, so this was starch hydrolysis. So how do we know that this is starch hydrolysis and not blood agar? The images that I'm putting aren't like meant to throw you off, like, oh, I guess it's just dark in that room. No, that's the actual color of the plate. I'm trying to help you out with that, okay? Is blood agar that dark brown color? No, it's like red. It should be. And so if it's red like blood, and then we would have clearing out of that, yeah, it would look pretty similar to that. But this was starch and it's dark because we had to add something to it, which I'm pretty sure is the next question. Starch hydrolysis, okay? If I think that there's going to be any confusion on something like that, I usually give some sort of hint or indicator for it, okay? All right, so which organism pictured is positive for starch hydrolysis? Excellent. So at the left, we broke down the starch. So now when we added the reagent, it doesn't react in that area and turn it that dark brown color. So it's just basically colorless. So what was that reagent that we used in starch hydrolysis to turn the media that dark brown color to allow us to see the halo? All right, so it's iodine. Or how much else to say about it? Okay. All right, which test is this one? Now look closely. All right, so that was nitrate reduction. We have two tests that can have Durham tubes, nitrate reduction and phenol red carbohydrate fermentation. The difference between them is the phenol red carbohydrate fermentation starts out that red coral color. Nitrate reduction is just like light broth color. So I guess you guys know what kind of questions are coming next. What do reagents A and B detect? I'll know you're gonna get asked about it on the test, so. Hey, A and B detect nitrite. Want to see what the other questions are for this one before I give too much away. A and B detect nitrite. It is a nitrate reduction assay. So what does zinc detect? Excellent, guys. Yes, you're killing it. Okay. I am so proud of you for this. All right. So if you can at least remember that zinc detects the nitrate, which you guys got this, and maybe the first one was a little bit of a hint, but zinc, the last letter of the alphabet, comes late in the alphabet. Zinc, nitrate, late. Okay. You got that? And you know that A and B is not testing for nitrate. Hopefully you can remember it's nitrite. So if we reduce nitrate into nitrite, A and B turns it red. If we don't, then we add zinc, right? Then if you change to red, then you're positive in that little zinc mini test because you're positive for nitrate still in the zinc test, right? Which means that overall in the nitrate reduction test, you are negative. Make sense? We're getting it, especially now that we filled out our forms, we started doing all that stuff. I'm sure it makes a little bit more sense, or at least for memorization purposes. Okay, what is detected at each step? First step, A and B, nitrite. Second step, zinc, nitrate. Red is not always positive for the overall assay, right? For the overall assay, the first step, red means positive for the whole assay. The second step, red means negative for the whole assay, right? And don't forget the gas in the tube. If you do have gas in that Durham tube, that would also mean that you were positive for nitrate reduction. All right, which test is this one? Proud of you guys. This is all one test, y'all. And y'all should know which one it is. This is the one, man. All right, so this is TSIA, triple sugar iron assay. All right, which of those tubes shows evidence of H2S production, dihydrogen sulfide? All righty. So that's going to be tubes 3 and 5. Why? Because they had the black precipitate. It's the same for TSIA as it is for the sulfur reduction part of SIM. Now I want to say, in the SIM test, the S for SIM stands for sulfur reduction. We also test for sulfur reduction in TSIA. It's the same thing. But which letter of TSIA is referring to that test? Do you guys remember? The I, the iron. So the iron is what's added to the media that binds the H2S and creates that black precipitate. So that one's not necessarily going to be a giveaway, but that's what we're testing for. But the product, the visibility of that black precipitate, that does tell you that it's sulfur. So remember that I means iron, and you should know that that also equates with the sulfur. Which tube in the same lineup shows evidence of gas production other than associated with H2S? So just 2 tubes for this. I feel like they're pretty obvious. They're pretty much screaming that they made gas. All right, so 4 and 4A. Those ones were lifted up almost out of the tube. That was the gas production from fermentation that did that. So TSIA, how does that get inoculated? This is the one that had the slant streaking and the stab, right? We get that stab involved because it pushes that bacteria down into the bottom where there's no oxygen. And so we want to encourage that anaerobic activity. All right, how do we read it? Oh my gosh, there's so many different ways. That's why we were going to have at least 2, probably 3 stations on TSIA alone, because you can have yellow for acid, you can have pink for alkaline, or you can have no change. And those things can happen on the slant or in the butt. You can have gas production, and you can have H2S production as well. Seems simple enough, right? Except now you have to know what those colors mean. So your slant is testing for sucrose or lactose fermentation. The butt is testing for glucose fermentation. So if you have a pink butt, did you ferment glucose? No, right? So that's the sort of thing that you need to be able to break down on the test as well. What colors are produced? We kind of went over that and what they mean as well. What is our pH indicator? Do you guys know what that one is for this? Phenol red, yeah. We only have 3, remember? Phenol red, bromothymol blue, litmus. Was it litmus? Absolutely not. Was it bromothymol blue? Absolutely not. So no, you have just to know 3 pH indicators. And then you just have to know whether your media has a pH indicator. So we look at the nitrate test. Did that have a pH indicator? So be aware of that too, because I might ask you what the pH indicator is on something that doesn't have one. Make sure you're paying attention, okay? All right, I'm not always nice, okay? So which test is this one? Uh-oh, this is now. So you guys are paying attention on the crap that I talk about. All right, so this is the urease test. This is the one we didn't do in lab because it wasn't made. And I said over and over again, it's going to have phenol red in it as a pH indicator. It'll turn bright pink if it's positive, and it's like a peachy orangey color. It's like a peachier version, right? So this is what our tubes would have looked like. And this is the picture that you have in your study sheet. I pulled it from the study sheet, okay? So if this appears on the test, it'll be this literal exact picture. This is what uninoculated urease looks like and what it looks like when it's negative. If it's positive, it's no question, okay? So I'm not going to give you one that's tightly, maybe you have to hold no. It's going to be these pictures, literally, if you see it on the test, okay? I haven't written the test. It doesn't take me very long. I just copy and paste questions from my bank. So I haven't picked my questions yet, but if urease is on it, I probably also gave you the answers to these questions too, which is fine. This is our practice with it. Nope, I didn't. That was it. Okay, which test is this one then? What's going on in this tube? There's always some people that get tripped up by this one. Don't get confused about which one is which. Always, this is the problem. So when you guys go into that test, you better have it clear which one is which. Okay, promise me. Because catalase, yes, because catalase catalyzes the bubbles to be made, okay? That's the reaction that we actually get to see being catalyzed by adding that reagent, which is probably the next question. Oxidase is a different test. In the oxidase test, we're testing for cytochrome C oxidase. This is a molecule in our aerobic respiration chain, that electron transport chain. It's the last one, gets that last electron, and it takes that last electron and gives it to the oxygen when we're getting rid of them. You know, how we have to get rid of them to oxygen. It's our final electron acceptor. Cytochrome C oxidase does that for some bacteria. Both of these assess aerobic, but one of them is associated with breaking down. What does it break down? The catalase? Oh. That's a question. What does catalase break down? It is called hydrogen peroxide, y'all.
Oh.
H2O2, hydrogen peroxide. If you guys, and it's literally in the name, you guys haven't noticed. H2O2. For every oxygen, per oxygen, there's a hydrogen, per oxide, hydrogens per oxide. Okay. Okay. I know, you never realized it before, maybe. I hate your chemistry teachers for that. Okay. Which test is this one? Oh, okay, now we should know this one. Now that we've cleared up the other one, the catalase. Hint, hint. Catalase breaks down hydrogen peroxide into water and oxygen. The bubbles that you saw in that test are oxygen. This purple, the indigo one, that one is our oxidase test. So that is a reagent you don't need to worry about what it is. But we added those little drops from that little baby bottle dropper, and it turned it either indigo or it didn't. If it turned indigo, you have oxidase, and that is cytochrome C oxidase present in the aerobic electron transport chain. All right, other tests that I don't have pictures of on here, but you need to know them and they are in your study sheet. Malonate, very similar to citrate, except it's broth. Green will turn blue if it's positive. Deaminase, that was phenylalanine deaminase. If you deaminated phenylalanine, the amino acid, removed the amine group, then when we add ferric chloride, that yellowy, oily drops to the slant, it turned the slant that green color. MRVP is two tests, the methyl red test and the Vogue's Proscar test. It's a broth looking broth. We split it up into those two tiny little tubes and added the drops. One had to sit for 30 minutes. The other one was instant. The instant one, we added methyl red. It turned red or orange. Red was positive for methyl red. That was heterolactic fermentation. VP, we added drops to that. It was one naphthol and potassium hydroxide. And if that turned red after 30 minutes, there was a layer at the top, but if it turned red at top, that was positive for mixed acid fermentation. Okay, so different ways to ferment and make acids. Gelatin, that was an easy one. That was to stab that butt, put it in the fridge. If it's still liquid, it broke down the gelatin. You had gelatinase. That's the enzyme that breaks that down. All right, let's go back to our bacteria basics. How would you classify this organism's characteristic arrangement? Not the one I want you to look at. Well, you guys got this. Another common problem that people have is not remembering the difference between strepto and staphylo. Strepto strips chains. Man, if you guys listened to that Friday review that I gave last Friday, where I realized that I put an example in there about strippers, that should help. Okay, so I'm just saying, strippers. Anyways, so this is strips or chains of those cells. They don't have to be cockeyed, right? So they can be strips of bacilli or chains of bacilli too. I just want to bring up really quick. If I am talking about the staphylo, I'm bringing this up because I had a question about this from a student last night. Remember, one of the augurs that we have to know about is the Staphylococcus augur, and it has salt in it. It doesn't have any differentiator in it, just the salt for selective. And my student asked me, well, if we're talking about mannitol salt augur, which we already know, being useful for identifying Staph aureus, why would we not also be using Staph augur to identify Staph aureus? My answer is kind of. My answer is this, not by itself. Because if Staph aureus grows on Staphylococcus augur, of course it would, because Staphylococcus augur, does only Staphylococcus grow on does only Staphylococcus aureus grow on Staphylococcus augur? No, any Staphylococcus can grow on that augur. Staphylococcus epidermidis, for example, one of the other common staph bacteria on your skin, which we learned about in chapter 19, right? So don't forget, Staphylococcus aureus is not the only Staphylococcus out there, not by a long shot. Those are just the only two we talk about in class, okay? So mannitol salt agar gives us more information. We have the salt to select for the gram positives, especially the staphylococcus. And then the fact that it breaks it down and turns it yellow, that's indicative of Staph aureus and not Staph epidermidis. So we can get that information from that agar with that one test as opposed to having to do more than one. Does that make sense? All right. I thought that if one person asked it, I should share it with everybody. All right, next question. What is the name and shape of these bacterial cells? All right, excellent. So that's bacillus. Bacillus are our rods. So we have the bacilli that are our rods, our cocci that are our spheres. We have staphylo, which is our grape-like or irregular clusters. And we have strepto that are our chains. Those are the only ones you're going to see on the test. You guys already know that. It's the only ones I have. So. All right, know your shapes. We just went through them, your arrangements. We just went through them. Don't get those confused with one another. Remember, a coccus is not an arrangement. It's a shape. And remember that simple stains are a thing. We've mentioned simple stains before. You're going to make your heat fix thing and then just stain with one stain. Helps you with contrast. And you can stain any cell, bacterial or eukaryote, with a simple stain. And when you do, it will be that color. So even a gram-positive bacteria, if you stain it with methylene blue in a simple stain, what color is it going to be? Blue. Okay. Gram-positive bacteria are only purple in a gram stain. That's it. All right, microscopes, easy stuff, should be anyways. How do we use it? All right, if you observe a specimen and your objective lens is at 100 and your eye lens, your ocular lens is 10, what's the total magnification? So we just multiply the two together, so we get 1,000. All right, which lens do we use with oil immersion? It's in the picture. That's the 100x. Other things you want to know about your microscopes, the parts of the microscopes. Know them, be familiar with the names and what they do for the microscope, right? We had the stage last time from the microscope. That was our part that I labeled. It's not going to be a stage this time, okay? Lab safety, general basics. Everybody needs to have a lab coat on the lab final day, okay? There might even be, I've done in the past where I'll have like one of the questions like not exist, and it's just whether or not you wore a lab coat, for example. So that's an easy one you don't want to miss. All stains, you want to know, we kind of already went over quite a few of them, but you want to know their primaries, any counter stains, whatever rinses are in the middle, and what those steps do for the entire process, what a positive and negative looks like. All of the tests that we are going to be doing for those tubes that we have, all the different media types that we have, those color changes, pH indicators, any reagents we have to add, and then be able to read an identification chart and use a dichotomous key. Those charts that were on the very first page of your unknown report. I'm going to give you one that's completely filled out, and I'm going to give you a dichotomous key, and you're going to have to tell me what organism it is. So I'm literally going to give you the chart. I'm going to say, what organism is this? And give you 5 choices. So you're going to have to be able to literally just look at that, the key. Oh, it's positive. It was cocci shaped. It was made urease, whatever. Okay. And I'll give you both of those though. You don't have to memorize them. You'll have the chart and you'll have the dichotomous key. There'll be two stations for that. One will be, 2 questions really. Question 49 and question 50. One will be about a gram positive. One will be about a gram negative. Okay. One of them, I might give you a chart that's filled out. Another one, I might just say, this thing had these characteristics. What is it? And it won't be on the chart. Okay. Like it might say that it fermented glucose. That's it. Or it was turned yellow. in the glucose fermentation test. That we fermented it, and you had to find that on, does that make sense? Just applying everything from all those tests all at once and having to find that. I try not to make them too overly complicated because I don't want to like double test you on things. I don't like that. I want to be sure you can use the key. So it's not meant to be like a trip up thing, but you do have to know how to apply it, okay? So that's that. That's the review. So let's see who did the best on it. Good luck studying, you guys. You did really good with the review. So honestly, I mean that. Sometimes when I do these reviews, I get so discouraged. But you guys do great. Now I gotta go found Dr. Rose for my letter. What?